Hi YouTube and Happy New Year. My name's Abraham Moeller, I go by John, and I'm a fourth year student in Emory University's Microbiology and Molecular Genetics PhD program. My dissertation research focuses on bacteriophages, or viruses that infect bacteria, particularly those that infect Staphylococcus aureus. I'm interested in phages as a way of improving treatment for infections caused by Staph aureus. Today I'm going to give a brief introduction to phages in general, but before I do that, I'll explain what's shown in these two pictures on the first slide. On the left is an electron micrograph of phages either attached or unattached to the surface of a bacterial cell, and you can see amongst those that are attached, one phage with a dark green electron dense core, and another one with an electron hollow, less dense, pale green core. The one with the dark green core still has the phage genome inside the head, whereas the one with the light green core has released it into the bacterial cell. On the right is another phage electron micrograph that's been annotated by an artist for its different components. The capsid, the protein coat that surrounds the DNA inside the head, is in pale blue. The tail fibers are in yellow, and then the base plate through which the phage DNA or phage genome is injected is in red. Before I go further through my talk, I'll give a quick outline. I'm first going to give an example of how phages can be good for us humans, and another example of how they can be very bad. I'll then briefly go through phage diversity in terms of shape and genetic material. I'll explain the two different phage life cycles and then two fundamental experiments from which you can learn basic aspects of phage biology. Now, like everything else in life, phages can be good and bad for us humans. They can be good in the sense that they help treat infections. The person photographed on the left is holding a vial containing a phage cocktail. This is a mixture of different phages that in this case are specific for intestinal bacterial pathogens. It's called the Intesti cocktail, and it was developed at the Eliava Institute in Tbilisi, Georgia, where phage research has been conducted for quite a long time. This has a major advantage over antibiotics. If you give these phages to a patient and introduce them at an infected site, just the infectious bacteria should be cleared. If you give antibiotics, on the other hand, that'll clear everything at the infected site, and that could be very bad. It could be very damaging to eliminate beneficial bacteria along with harmful ones. Now, phages can also be bad from a human perspective. They can be bad in the sense that they interfere with industrial microbiological processes that depend on bacteria. One example is yogurt culturing. This depends on lactobacillus to culture the yogurt. And at an industrial scale, phage contamination can reduce culturing efficiency, and instead of getting nice viscous yogurt out of this, you get this milky watery stuff. Quite unpleasant, quite wasteful. It turns out CRISPRs, now famous for gene editing and various kinds of genetic engineering, was first patented as a way of improving yogurt culturing efficiency. Bacteria containing CRISPRs would be resistant to phage and thus robust to phage infections, and you'd get nice viscous yogurt instead of milky watery waste. Phages are highly diverse in terms of morphology and their genetic material. Again, viruses are the most abundant life forms on the planet. There's quite a few different kinds of phages. These can be non-enveloped or enveloped, so just a protein coat or protein surrounded by a lipid bilayer. They can have double-stranded DNA as genomes, single-stranded DNA, and double or single-stranded RNA. Despite this, probably the most famous phages are the cauda-virales, or tailed phages. A prototypical example is shown in the upper right that looks quite like a lunar lander. On the left is a two-dimensional cross-section, and I'd like you to focus on the head. you can see protein that surrounds the DNA inside. On the right is a three-dimensional representation of this prototypical Cotta-Virales phage. The head's on top, that's icosahedral, so 20 faces that are triangular. There's a collar linking that to the tail that's cylindrical, the base plate under that, and then the tail fibers that extend from the base plate, much like the legs of a lunar lander. Even within the Cotta-Virales, though, there's even more morphological diversity. The three main classes of the Cotta-Virales are the Myoviridae, the Podoviridae, and the Cyphoviridae, which have a long contractile tail, a short non-contractile tail, and a long flexible non-contractile tail, respectively. The Greek here is helpful for memorizing these classes. Myo for muscle, for contractile, podo for foot, that connects with short, and then cypho for tube, so flexible. Phages have two different life cycles, the lytic and lysogenic cycles. Phages don't necessarily cause lysis, they don't necessarily explode the cell after attachment. But when they do, this process starts with the binding of the phage to the surface of the cell, and then the step called entry. This is when nucleic acid is injected from the phage into the cell through the tail. After this step is transcription, translation, and replication. The phage genes are transcribed and then translated into proteins, and the phage DNA, the phage genome, is replicated, so there's multiple copies of the phage genome and proteins that are important for putting the phage together. The next step is then assembly of the phage and packaging of the nucleic acid into these new phage particles. And once that's completed in a coordinated time step lysis, the bacterial cell is burst open and phages are released that can infect other cells. As I said earlier, phages don't necessarily burst the cell after attachment. They can also undergo the lysogenic cycle, in which they enter a latent state. The first step of this process is repression, in which a phage-encoded repressor turns off the lytic cycle genes, and then integration, in which the phage genome is integrated into the host chromosome at what's called an attachment site, an at B site on the bacterial chromosome and at P. There's recombinases that will recombine the DNA and thus integrate it into the host chromosome. The new cells, the lysogen containing this prophage, can replicate and carry this latently infected phage, and then upon stressful conditions, the phage can be induced to re-enter the lytic cycle. What are some examples of stressful conditions? One is DNA damage caused by UV treatment, another one's nutrient deprivation, say a starvation of amino acids, for example. A major question in phage biology is, why have these two life cycles? Why have lysogeny at all? Well, it could be advantageous to exist in a latent state in certain environments and only enter infection when the host, the bacterial cell, is a sinking ship during those times of stress that I mentioned earlier. Now, I want you to think about this lytic cycle when I go through this first of the two experiments I'll describe, the single-step growth curve. This is for studying how phages grow. And the way you do it is you take some quantity of bacteria, in this case E. coli B, and infect with an equal quantity of phage, in this case phage T2. Over time, you measure the concentration of phages released into the culture. During a certain period called the latent period, there is an interesting observation that you'll make. No phages are released. There's no change in the plaque-forming units, this measure of phage concentration in the culture over time. It's only at the rise period where lots of phages are released and then this process ends. And from this procedure, you get a measure of the kinetics of phage infection, these two parameters, the latent period and the burst size, that tell you the kinetics of phage infection. And these vary between different phages and from host to host and between host mutants where the mutations might affect the phage infection process. I want to point out one other observation in this graph. I mentioned there's this latent period that here is about 20-something minutes before phage is released, but there's also an eclipse period much shorter before which you don't even see phages inside the cells. So there's some period before which you don't even see phages inside the cells. It's only after that that you notice it, say, by doing microscopy. Now I want you to think about those observations and that phage lytic cycle I just described. Why is it that there's no phage released for some time, unlike bacteria where they're just doubling and doubling and through the log phase growing incredibly quickly? Well, if we think back to the lytic cycle, there's lots of steps that have to happen before phages are released. Lysis is not an immediate process. There's enzymes important in entry and in coordinating transcription, translation, replication of the phage genomes, and then assembly of the particles before lysis can even happen. And that's why you see no phages produced, and you see these stepwise staggered dynamics as opposed to a continuous growth dynamic as you would see in bacteria. This is a really important fact about phages and viruses in general when you think about their replication cycles and their populations. Now, a second important experiment in phage biology is just measuring phage concentration altogether. Say you have an environmental sample and you want some estimate of the number of phages present that infect E. coli. Well, you need to do a phage titer. And how do you do a phage titer? Well, much as you would measure concentrations of bacteria, you take a phage stock and you do a serial dilution in sterile medium. In my case, I use something called TMG that buffers pH. It has cations that are important for phage absorption and stabilization, and then gelatin that also helps with stabilizing phage. In this case, you're doing a 1 in 10 dilution, so be sure and change your tips with each dilution. And you proceed doing your serial dilution, transferring volumes, and then you take some volumes and you plate them on a permissive host and look for plaques. What's a plaque? Well, a plaque is an area of clearing of cells that starts with a single phage infecting a single bacterium. So you imagine you could get an estimate of plaques per volume from these, phages per volume from these plaques that form. A single phage infects a single cell. That cell bursts and releases phage that infect neighboring cells, and the process spreads throughout. On the right is the result of one of my experiments. Instead of plating volumes on individual plates, I've put spots of different dilutions on the same plate. I get to a point where there's countable dilutions. Once I've counted the plaques, I divide that plaque count by the dilution factor and the volume plated to get an estimate of phages per volume. And with that, I hope you've learned more about phages from this video and that it was useful to you. I appreciate your attention. Please like, comment, and subscribe. If you have any questions, let them know in the comments section. Be sure to check out my other videos posted on this panel, and thanks again for watching.